Everyone, this is Yongting. I will be presenting the paper on pheromone. So what is pheromone? Pheromone is body secreted fluid that can be detected by the individual, and it can also affect neuroendocrine brain and its behavior. In majority of the mammal, it's induced in the VNO and access through olfactory bulb, go through media and amygdala, and then go through anterior hypothalamus. And anterior hypothalamus is well known to influence sexual behavior and reproductive function in a sexual specific way. In today's paper, or any paper they mentioned, they have concluded that study have not shown there's any convincing pathway for people to understand the connection from VNO to brain in humans. So in their study, they are creating pheromone form steroid that called AND and EST. I will go into it more, but let's introduce the scientist that's really critical for one of the paper called Descent Sexuality, which I'll talk about later, and two other authors will be covered by my group mate, Mark Brilov. He earned his bachelor degree in psychology from Yale University in 1976 and PhD in psychology in 1992 from UCLA. He was a professor of psychology at University of California, Berkeley from 1982 to 2003. Now he is a professor of neuroscience at Michigan State University. He works in the field of behavioral neuroscience and neuroendocrinology. He also demonstrated that steroid hormone and sexual behavior affect the developing and adult spinal cord and brain. In the first paper, it's more like a neuroscience background, so I want to introduce the two sexual hormone they are derived with. One is called AND, is derived of testosterone produced in human. It's secreted 20 times higher than in men than women, and EST, naturally occurring estrogen. They have asked 24 healthy subjects divided evenly between female and male to do a measurement of regional spiral of blood flow using PET scan during passive and bilateral smelling of AND and EST. Because AND and EST is reported to change in mood and skin conductance and heart and respiratory frequency in sexually differentiated manner. So basically what happened is in female, when they are letting females smell AND and EST, they found that AND activate the anterior ventral hypothalamus, showing figure 1 and 2, but not olfactory region. And using AND versus air, they showed that there's a cluster in the right amygdala, and activation have been shown in ventral medial nucleus. And they have used volume of interest analysis and random effect analysis to confirm the result that hypothalamic activation with AND was significantly higher in the female. And in the male, they found that activation of hypothalamus only when males smell EST but not AND, and EST versus air, which is as control, showing there's a cluster in right and left of amygdala. So in the male, they found dorsal media and periventral nucleus has the activation when it's smelling EST, which also has been confirmed by volume of interest analysis and random effect analysis. From there, they are asking the question, which is do AND and EST activate the human brain? In the study, it has shown that both AND and EST has activated sexual specifically irrelevant of hypothalamic region. And they also want to know, are these activations sex specific? As I mentioned, AND and EST activate different region in the male and female brain. And as we can see, AND activate female brain and EST activate closely in male brain in the hypothalamic region. And all those activation, it's mediated, connect to the mediated reproductive behavior. We want to confirm that the observed hypothalamic activation is truly sexually dimorphic because there's significant sex VIO interaction of AND and EST activation. They also have mapped out different pathways to suggest why that it is. They think that hypothalamic pathway may be direct from olfactory bulb and separate from the bulb olfactory pathway. Basically, what they're trying to say is there might be two different pathways in the brain. That one, it's specific control the hypothalamic region in sex like perfume, and the other one is more passive odor, which they have tried 60 different odor that's non-AND or EST, and it's going through activation of olfactory region inside hypothalamus. So hypothalamus is really specific to AND and EST, which is sex specific. In the next paper, it's continuation of this paper. Basically, they do a brain response and think testing the pheromone of AND and EST, but in homosexual male, and compare it relative to heterosexual female and heterosexual males. They are looking more into hypothalamus because there's many research have been done that lesion in anterior hypothalamus or impairment of pheromone production can alter the mating choice and its behavior. So in animal, we did know that pheromone go through the VNO and it goes through olfactory nerve, which eventually reaches to the hypothalamus, but in human, these kind of mapping mechanism of the pathway is still unknown. So in this paper, they want to know like the pattern of activation induced by AND and EST to confirm it's relatively explaining is this sexual orientation or biological sex. So in the study, they recruit 36 healthy unmediated right-handed HIV negative heterosexual men, heterosexual women, and homosexual male, and 12 in each group. And the result is they using the ROI analysis and saw that there is different pattern appear in the homosexual man. So as before, we know that heterosexual women show activation of anterior hypothalamus with AND, whereas heterosexual male has been recruited by smelling EST. we might expect the same thing happen to the homosexual males, since due to their biological sex, we're expecting that they might have same activation as heterosexual male, but actually they find out through the scan that in the homosexual male anterior hypothalamus, it's only activated with A and D, just like heterosexual women. So when homosexual males smell EST, the left amygdala, which is associated with olfactory, is recruited. They are also using 4 different smell during the scan in contrast of AND and EST, and they still would find the consistent pattern that the homosexual male show pattern of activation resemble of heterosexual women rather than heterosexual men. And that makes sense because this also leading to the conclusion where they think that your sexual orientation for like heterosexual women more likely is opposite sex, so they attract to men, which consider as A and D, and homosexual men also attract to men, so they also are attracted by smelling the A and D. The difference that we see between the homosexual men and heterosexual men could reflect their deviation of anterior hypothalamus in homosexual men. That's why they use 1 group random effect analysis and 2 group random effect analysis to confirm the result, and from both results, they see that only smelling A and D will lead to the hypothalamus activation in homosexual male comparing to the heterosexual male. They also use other tests such as post hoc tests to analyze the result relative to the air in the separate group, and they find this to be consistent. Basically, they think that there's activation for AND and EST in a separate group of subject, and they think AND and EST might be partially overlapped due to their activation in amygdala, perform cortex, and minor portion of anterior insula. But most importantly, there's a suggesting that the preferred pathway in the present compound is associated with the subject's sexual orientation, at least in this case, investigating in men rather than their biological sex. There's other paper that my group may will talk about in the lesbian or transgender. They find similar consistent results. And they also confirmed that co-localization of hypothalamic response with brain circuit are involved in human reproduction, which that animals are designed to recognize the sex further indicated by the hypothalamic involvement in the psychological process relative to their sexual orientation in human. At least from these 2 papers, we're confirming the activation in the brain, such as hypothalamus, is directly linked to sexual dimorphism and their sexual orientation. The next paper is the one that I really got interested because it explored the reason why we are seeing those same sexual behavior, what now they call homosexuality. And this paper go into gene, like the mechanism they suggested that might contribute to the phenomenon we're seeing in modern day. So they start with exploring different species, and the species that human are in somehow has a dysfunctional gene called TRPC2. More importantly, they also explaining what is TRPC2. In mice, where they start doing the study is in mice, the disruption of the TRPC2 gene, that it's really crucial for transducing the chemical sensory signal from pheromone to VNO. When they do some manipulation by causing dysfunction in the gene, it increases the likelihood of SSSB, and the loss of that function lead to the loss of sexual dimorphism in adult brain in mice. So they're suggesting maybe back then the TRPC2 appeared to be relaxed, causing gene accumulation to prematurely having stuck colon or frame shift or missense mutation that lead to the dysfunction gene, which we think happened in human around 25 million years ago. More importantly, long time ago in the ancestral primate, the VNO still exists. It has a signal through TRPC2, and this is where they promote sexual specific brain organization to address sexual behavior, almost to like just exclusively opposite sex partner. So long time ago, we are only absorbing opposite sex behavior, but now when they're exploring and doing a knockout mice, by knocking out the TRPC2 in the mice, they found that it's not like they're not able to discriminate male and female concept. It's they're rather having the attraction toward the same sex, which what we see as very virtuous same-sex behavior in mice, and extend that to humans. They're assuming we also lost a lot of people have just through ancestral genetic tree that it lost TRPC2 gene, and that causing the onset of SSSB converge in our lineage. And they are not sure what's the reason, but they are confirming that the loss of the protein previously have enforced the opposite sex sexual behavior by organizing or activated brain response to the male-female pheromone, more likely for primate to increase the behavior of having homosexuality. And they assume maybe around like 25 million years ago, there might be some loss of function of TRPC2 lead to like advanced brain development, which might be beneficial for their survival, to have preference for same-sex partner in some individual. So that mutation just got reproduced over and over and extended to our lineage. So the takeaway from this paper, it's TRPC2 gene is really critical and it's dysfunctional in human. That's why it leads to higher possibility of the same-sex sexual behavior we observe in modern day has been long inherited from our common ancestor. And I have concluded the summary from all 3 papers here. And thank you for watching.